Welcome to week 1 for Hematology 2. We are going to go through 2 chapters this week, so let's just jump right into that. Let me share my screen so I can pull up the PowerPoint. All right, so the first chapter is going to be titled Hemoglobinopathies. It should be chapter 27 in the latest edition of the textbook. So hemoglobinopathies, and then the next chapter will be thalassemias. So we are going to go through, I'm not going to read through the objective slides as normal. You can always, read those on your own. The objectives, again, are just to kind of give you an overview of what you should be really getting out of the chapters. There are a lot of objectives for these chapters. There's a lot of information in these chapters. I'm trying to whittle it down so that you don't have to feel so cumbered down. These are some tough chapters, I will say that. I think they can be really tough to learn. So I will make sure, as always, when I lecture, to point out the things that I find very essential, things that I would definitely take away from them. And then you can look at your review questions that you do for lab to help you look at the chapter information on those specific areas that I really highlighted, and that should all help you get through it. All right, so overview. What does the hemoglobinopathy mean? Basically, when you look at the word itself, we obviously see the part of hemoglobin, so we know it's something to do with hemoglobin. The part pathy always refers to a disease. So when you think of like a pathologist, that's a doctor who studies disease. Whereas hemoglobinopathy means it's going to be a disease involving the hemoglobin molecule. So something in the way that hemoglobin was made is wrong, is mutated, it's not correct. As a result, it makes abnormal different hemoglobins, which will mess with how we deliver oxygen and all that kind of stuff. So there are actually 2 overall categories of hemoglobinopathy. There are qualitative and quantitative. You do need to know these categories, what they mean. So this slide to me is really important. I would highlight this and take it away. Qualitative is essentially meaning that you're making a normal amount and all that, but the way that it was made is abnormal. So remember, when we make a hemoglobin molecule, it was made up of 4 heme groups and 4 globin chains. And those globin chains were strings of amino acids put together. That's what makes it a protein. So somewhere in when they were making those globin chains with the amino acids linked together, they were in a set order, something in the amino acids got changed. So there was an alteration in the amino acid sequence in the globin chains. That is a qualitative hemoglobinopathy. So because one amino acid got put in and another got taken out, it changed the entire hemoglobin molecule then. So that would be a qualitative. A quantitative is just like it sounds. It's involving an amount, a quantity. So the way that you're making the hemoglobin molecule is okay, but you're not making enough of it. It's a lack. So what it says here, a reduced rate of hemoglobin synthesis. The amino acid sequence is fine, everything there is okay, but you're just not making enough. So that is the 2 broad categories. So when we apply it to what we're going to discuss here, in this chapter, PowerPoint, we are going to go through the qualitative ones, the ones that are what we call structural defects, in which the amino acid sequence did get changed. The quantitative group is the thalassemia group, and that's going to be in the next chapter. It should actually be chapter 28 in the new edition, so you can ignore that it says chapter 27 there. It's 28. So that'll be in the next chapter, the thalassemias. Truly, when you think about hemoglobinopathies, the true category they belong in would be more the structural defects. That's the disease part. But thalassemias are definitely still involving hemoglobin molecules, and those would still be a quantitative lack of them. So we'll learn that in the next one. All right, so overall, there are 6 globin genes on 2 different chromosomes, chromosomes 16 and 11, that are responsible for how your globin chains are being made. Something in these genes have probably been mutated to get a structural defect. So somewhere in these genes, something got changed or mutated, and that's why all of a sudden you'll get an abnormal hemoglobin showing up and a different amino acid sequence. Now, you do not need to remember these chromosome numbers. This is not something you have to remember. This part was just informational. So let's review this lovely little chart. We had this chart back in Hematology 1. And so, again, Every hemoglobin has 4 globin chains in it. And those 4 globin chains are what determine what type of hemoglobin you have. That's kind of how we name the hemoglobins. So if you go all the way down to the last row, the adulthood, those were the ones that we had to remember from hematology 1. That 2 alpha, 2 gamma were hemoglobin F. 2 alpha, 2 delta were A2, hemoglobin A2. 2 alpha, 2 beta would be regular hemoglobin A, which is the one that we have the majority of. At birth, when there's a newborn, they have majority hemoglobin F at that point before it switches. And then if you go up the chart a little bit more, you'll see the intrauterine. So while they're, in the, amniotic sac and all that, there's different types, Gowers and Portlands there that are involved, but they don't stay involved after a certain while. So they kind of go away after a while and it changes. But this is overall what a normal kind of process looks like from all the way through different developments. So there are actually greater than 800 structural hemoglobinopathies. That's a lot. That's a lot of different hemoglobin diseases, different amino acid sequence switching. Now, we are going to whittle that greater than 800 number down to just a couple that I really, really want you to remember out of all of them. And so we will go through them. The way that most of these mutations are occurring, again, it's a genetic mutation in some of those genes. Point mutation is probably the most common type of genetic mutation that occurs to create an abnormal hemoglobin. And that just means they're literally taking one nucleotide, one of those amino acids out, and putting a new one in. Not amino acid, one of the nucleotides in the gene and putting a new one in, and that's what mutates the gene. And then that mutated gene will create a new amino acid sequence that shouldn't have been there. So that kind of just all goes hand in hand. But overall, again, this is more informational here. We're just going to have a few very common ones that we're going to go through. So there's overall the other types of genetic mutations that could occur to result in these mutated genes that make mutated hemoglobins. Again, all just informational here, nothing I'm going to test you on. So overall, the pathophysiology, what that means is the background on what happens when you do this. When an amino acid is substituted, you will give a different charge to the hemoglobin molecule, and the size of it will change because this will change the protein itself. I mean, hemoglobin is a protein. So when you substitute an amino acid out and put a new one in, it changes the whole look of it, the whole feel of it. So where in the amino acid substitution got taken place, it will vary on how significant that is of a change to the hemoglobin. Some substitutions are more significant than others, so it's just that will make the disease vary from more mild to more significant, basically. The most frequent hemoglobinopathy that you and I will discuss and that you will see is going to be hemoglobin S. So at the very bottom there, it says the most frequent abnormal hemoglobin and the most severe is hemoglobin S. So that is what is responsible for sickle cell anemia. So that's what we'll kind of get into when we first get into these here. We'll start with sickle cell anemia. So overall, they've kind of classified these hemoglobinopathies into 4 basic categories. The first is an abnormal hemoglobin that results in a hemolytic process, so sickle cell anemia would fit in there. 2, there is a specific one called methemoglobinemia, or they can nickname it hemoglobin M. Third category is hemoglobins that have increased or decreased oxygen affinity. And then finally, 4th, abnormal hemoglobins that just don't have a ton of clinical effect on the patient. All right, so let's begin with sickle cell anemia. It should go without saying, if this is the most common one and most significant, you need to know it. So basically, in sickle cell anemia, instead of having all those normal hemoglobin A, A2, and F, they have hemoglobin S as the majority of their hemoglobin. So what created hemoglobin S is the second bullet here. There was an amino acid substitution. It was specifically glutamic acid being replaced by valine, and it's on the 6th position of the beta chain. So in that beta chain, where the 6th spot was, it should have normally been glutamic acid, and instead, because of some gene mutation, all of a sudden valine was put in place of it. And so it changed the entire charge and the size of the hemoglobin to make this hemoglobin S. You have to know that amino acid substitution. You have to memorize that. It's simple as that. You need it for this class. You need it for your boards. So it's really important that you know that amino acid substitution. Now, that sickle gene that was mutated was inherited from one parent. And then if that parent joined up with another person that had it as well, both parents have it, that's how you get sickle cell anemia. Now, it could be that maybe just one parent had it and the other didn't, and maybe it would be a sickle cell trait, like a carrier, and we'll see that on another slide here. Or if you got lucky enough, one parent had sickle cell gene and the other parent had another type of hemoglobinopathy gene, then you can make a combo effect. So there is that possibility to see this in combination with other things, not just hemoglobin S. So, you know, it depends on the fortune, I guess, that you had of your parents. Basically, most of the time where we see sickle cell anemia is it's in pretty set areas. It's mostly African descent, Middle East, Indian, and Mediterranean heritages tend to have these genes passed down. So if you have any sort of, you know, if you have any sort of Indian or Middle Eastern or anything like that within you, you know, we're all made up of different things. You could have maybe a gene within you that you've been carrying and don't even know it. And then if you were to have, you know, a child with another person that had it as well, that could eventually result in sickle cell anemia. So, I mean, you guys get how genetics kind of work. But at any rate, so need to know the amino acid substitution. The other neat, well, if anything can be neat, it would be this, which is the first bullet point there, that there is actually a natural resistance formed against Plasmodium falciparum, which of course is malaria. We've talked malaria last quarter. So hemoglobin S has this natural kind of resistance against that Plasmodium falciparum parasite. Remember, those parasites use the red cells to their advantage. Well, they kind of have this resistance against it, which is good because when you look at down at the bottom who this mostly occurs in, it's in the same areas of the world as malaria. And so it's kind of one of those things that just through evolutionary time has happened. You know, other than that, I don't know a ton about it, but, you know, keep that in mind. That's a good point to keep in mind there. All right, so when we see that amino acid substitution take place, like I said before, it changes the charge of the hemoglobin. So in this case, it will turn the hemoglobin to a +1 charge. So that will change the structure of that hemoglobin molecule. As a result, it will affect the amount of oxygen it can carry. So what happens is, in order, you're probably thinking, well, where do the sickle cells come in? And this is how it occurs. And it all is from that hemoglobin amino acid sequence change. So the sickle cell will occur when your oxygen saturation on that hemoglobin molecule drops to less than 85%. So if they're carrying less than 85% full capacity oxygen, the cell will begin to sickle in shape and get those crescent moon pointy ends kind of look. So in other, you know, so it doesn't have to be always less than 85%. The number will vary depending on if it's homozygous, heterozygous, that kind of thing. So that is how kind of the sickle cell part of it comes into play. Now, there are sickle cells that you might see on the smears. Sometimes you might not see them, but, you know, just because you have sickle cell anemia, you usually will see them, but not always. Just depends on if they're reversible or kind of irreversible in regards. Like I said before, the asymptomatic fatal just depends on the different combinations you have. If it's hemoglobin SS, that would be the homozygous. That's probably the more significant one. Then there's a combination of hemoglobin S and hemoglobin C. Then there's combinations with thalassemia. So it just depends on what is comboed up there. The hallmark clinical feature of sickle cell anemia is something that we call vaso-occlusion. So it's bolded there for a reason. Out of this slide, that bolding is important. So here's what vaso-occlusion is. On normal red cells at the top, you can see they're going through the blood just fine. They're passing through your, veins and arteries okay. When you have somebody with sickle cell anemia with hemoglobin S and that sickling starts to happen, you can see by the third step there, those sickle cells. Do you see how that starts to block and occlude? And then it will go even more, blocking and occluding. And that's what vaso-occlusion is. It's the blockage in your vessels, and it's very painful to that patient. So this side part just talks about in a red cell that has undergone repeated sickling episodes becomes stiff. Non-flexible red cells are caught up in the microvasculature, and they get stuck. So as these cells sickle and then become normal, sickle and become normal, they get really stiff and they lose that flexibility. That's kind of the hemolytic process as well. Sickle cell anemia is a hemolysis occurring as well. It will cause lysing of the cells because they're stiff. They're not going to be able to transverse through small spaces. So it says red cells with hemoglobin S behind the non-flexible red cell give up their oxygen. So again, as it loses the oxygen saturation, drops to less than 85%, it will sickle. And then as it sickles, it will form a blockade. So that's just what it's showing you. That's what vaso-occlusion really is. That's where the hemolysis part comes in and all that good stuff. All right, so let's talk about lab world diagnosis. Again, you will see it's a hemolytic anemia. When we see hemolytic anemia, remember, we would see schistocytes on the blood smear. We would see increased reticulocytes because you're trying to replace those destroyed cells. And then, of course, for sickle cell anemia, you're going to see definitely sickle cells, otherwise known as drepanocytes. Target cells are a big finding here as well. Kodocytes, what we call kodocytes, we see a lot of that. Nucleated red cells, that is saying that we're seeing immature red cells, and that's because, again, it's a hemolytic anemia. We would see that those red cells trying to get pushed out of the bone marrow to replace the destroyed red cells. And then a few other things like basophilic stippling, Pappenheimer bodies, and Howell-Jolly bodies are all seen. So this is a really important thing to remember, all those good things that you see of poikilocytes and inclusions. Marked anisocytosis, so that's saying lots of variation in cell sizes, increased retics. Again, you're trying to replace the hemolysis. As far as screening tests go, if you see all this kind of stuff, and by the way, this is a normocytic, normochromic anemia. It's within the normal reference range of size for the normal red cells. It's MCV and MCHC are normal. But if the doctor were to see like all these other things, they might say, let's do a screening test. Maybe this person does have sickle cell anemia. So a screening test for it would be hemoglobin S solubility, and it would be positive. So there's hemoglobin S is insoluble when it doesn't have oxygen attached, when it's in that sickle shape. So we call it a hemoglobin solubility test, and it would be positive. The minute it's positive, it should reflux into an electrophoresis. Electrophoresis is going to be our answer to diagnosing all of these different hemoglobinopathies. Remember, electrophoresis is basically taking proteins and moving them across an electric field, and the proteins will separate, and so you can kind of see what type of proteins you have. Well, hemoglobin's a protein, so if we take that sample of blood and move it, we can see the different types of proteins we have. In normal, healthy people, they should have hemoglobin A, A2, and F. In somebody with sickle cell anemia, you are going to see greater than 80% of their protein, of their hemoglobin, be hemoglobin S. So that would confirm, yes, you do have sickle cell anemia. So electrophoresis is key to confirming the majority of these hemoglobinopathies to know exactly what types of hemoglobin are being shown. So hemoglobin solubility screening test positive, and then hemoglobin electrophoresis would confirm it with showing greater than 80% hemoglobin S. All right, now I did say that there's like a carrier state, or what we could call a sickle cell trait. Again, it's a benign, asymptomatic. The patient probably doesn't even know they have it unless they've been tested or something for it. So they don't even know they have it. They're totally healthy. They're going to have normal red cell morphology. I know it says you might even see a few target cells, but I would just really go with there's no target cells on there on the smear. So I would kind of keep that in mind. Really, there's not any target cells on the smear. They will still be able to do a positive hemoglobin solubility test, and that is they are a carrier. They do possess some hemoglobin S because they're like having the trait that's like a heterozygous, if you will, state. It doesn't affect them because they still have the majority of the normal hemoglobins, like hemoglobin A, which can do the job and it's fine. But because there's still some hemoglobin S there, that will still cause for a positive hemoglobin solubility. And then if you were to do the electrophoresis, you would see around 40% S. And then, again, the majority of the rest is A, as always. So that kind of tells you it's trait and not the full anemia. Okay, next hemoglobinopathy would be hemoglobin C. This is probably the second most common hemoglobinopathy, pretty much just exclusively in the African American culture area. Again, amino acid substitution is responsible for this one, and you do need to remember this amino acid substitution. In this case, it's lysine being substituted for glutamic acid, and it's actually in the same location as the S. It's in the 6th position of the beta chain. So both hemoglobin S and hemoglobin C had a substitution in the 6th position on the beta chain. In this case, it's lysine being put in. There should not be lysine there, but that's what's getting put in there. So it will change the charge of the hemoglobin molecule to a +2 on this one. Now, as a result, there is this thick crystal that can form in hemoglobin C disease that we call hemoglobin C crystal. If you guys think back to that poikilocyte inclusion study guide that we did, that should have been on there, and you should have had that on there. It's like a thick rod shape. So we would see that on a smear with somebody with hemoglobin C disease, so keep that in mind. Again, it's still a hemolytic kind of process, so you would still see increased reticulocytes and then definitely increase in target cells. Well, you should never have target cells, so target cells would be present. Target cells are kind of seen through all of these hemoglobinopathies. That's always kind of a key finding. When you would do the hemoglobin solubility test, it would be negative. It has a different amino acid substitution, so the solubility is different. And then again, to confirm it, you would do hemoglobin electrophoresis, and then you should see hemoglobin C is on the electrophoresis pattern. All right, so that's what the hemoglobin C crystal looks like, that thick, short rod there. All right, there are a couple combination ones, not combo, I should say double substitution amino acids that can happen. There's one that's called hemoglobin C Harlem, otherwise known as hemoglobin C Georgetown. In this case, they have the substitution of valine for glutamic acid, which is the same substitution that hemoglobin S had, plus the aspartic acid substitution for asparagine. Seems like asparagus, doesn't it? And I'm the only one that probably laughed at that. So in this case, there's 2 substitutions happening. So this one will migrate with hemoglobin C on electrophoresis, that's kind of the naming of it. If you were to do a hemoglobin solubility test, actually it would be positive, and that's because it possesses the same hemoglobin S substitution. So that caused the whole insoluble nature. Since it has that same substitution, it would still cause a positive solubility. But again, the electrophoresis would show that it's not hemoglobin S, but in fact it is different. All right, hemoglobin E. I do find this one important. I would ask that you remember this one as well. This one tends to occur more in the Southeast Asia culture, 30% around. The amino acid substitution, again, is happening in the beta chain where lysine is being subbed for glutamic acid, but it's in a different spot. It's not the exact same as hemoglobin C. It's in a different location. The hemoglobin solubility test would be negative here since it's a different substitution that's not insoluble. And again, you would just confirm it with electrophoresis. Hemoglobin O Arab, the name kind of tells you the nature of where it's found, Kenya, Israel, Egypt. Many target cells, again, there's target cells on all these. And then it shows the amino acid substitution. I am not going to ask you to remember this one really, so don't worry about it. All right, here's one of those combination where you have both hemoglobin S and hemoglobin C happening. This makes a unique crystal called a hemoglobin SC crystal. And when you look in this picture, I'm hoping that you can see my arrow here. I'll circle around the one. Here's one of the hemoglobin SC, and then up here is another of those crystals. They aren't always that easy to see or recognize right away. I always think of them as kind of like finger projections. They kind of have different patterns they can take on, but like if you were to make the Y with your hand, that's kind of what it looks like on the one, you know. So they kind of have, they're not as nice and easy and predictable. They have a little bit of different shapes, but that is known as the hemoglobin SC crystal, and so that is seen with hemoglobin SC disease. Positive hemoglobin solubility test because hemoglobin S is present. And again, when you go to electrophoresis, you would see both hemoglobin C and hemoglobin S migrating to know it's the 2 of them together. All right, hemoglobin M is methemoglobinemia, methemoglobin in other words. Now, we learned about methemoglobin in Hematology 1, and some of you may not remember it because it wasn't like discussed in depth all the time, but it was back in the hemoglobin chapter 10, it should be in the book, unless that number got changed, but it should be the hemoglobin chapter. We discussed it. Hemoglobin M is when your iron changes. Remember, in nice, healthy hemoglobins, it should always be in the ferrous 2+ state. When iron changes from 2+ to 3+ ferric, that's what makes methemoglobin. So that's kind of what goes in these blanks right here. When your iron oxide goes from 2+ to 3+, that will create hemoglobin M or methemoglobin. Remember, when we learned about this back in chapter 10, we learned the problem with methemoglobin with the iron change is it won't bind that oxygen. It's like, I ain't taking it, and thus your tissues don't get the oxygen it needs, resulting in cyanosis. So it just kind of is a tie-in there. It's still a hemoglobinopathy because it's an abnormal hemoglobin. All right, other unstable hemoglobin variants. There are different ones listed at the bottom. The most prevalent one is probably hemoglobin colon. Otherwise, there's a bunch there kind of listed. So I'm not going to say too much on that. I'm not that worried about this. Hemoglobins with increased oxygen affinity, meaning that they love their oxygen so much that they will not deliver it. So these would just be anything that really, any hemoglobins that will alter, that change the way that they will deliver the oxygen. They keep hanging on to it. Again, it deprives your tissues of oxygen, resulting in hypoxia. Remember, when your kidneys sense that hypoxia, the kidneys will release erythropoietin, which is that hormone that goes to the bone marrow and tells us we need more red cells produced. So that whole system will be there in place. So usually it's asymptomatic. The patient probably isn't aware unless there was some testing done at one point for it, but because the kidneys just up the erythropoietin, get more red cells put out, we're not even aware that there's as much, failure to release the oxygen. Usually your compensation is working for it. All right, so that is it on hemoglobinopathies. I think the big ones out of here are going to be hemoglobin S, of course, sickle cell anemia, hemoglobin C, hemoglobin E, and then it would be just knowing the categories of the hemoglobinopathies, qualitative versus quantitative, some of that stuff I said in the beginning. So, there's a lot more hemoglobin changes out there, but we just can't go through them all. And most of them are pretty rare. All right, let's jump to our next chapter, which is chapter 28 on thalassemia. All right, so we'll just skip through the objectives. So again, thalassemias are in the quantitative category. And there is some alteration to their gene, but it causes a decreased rate of hemoglobin synthesis of their globin chain. So it's still affecting the globin chain, but the globin chains are made normally when they are made. It's just that we're not making enough of them. It's a decreased rate. So that alteration or mutation that happened to the gene changed how much we're making of those globin chains. You don't make enough globin chains, you can't make hemoglobin properly, you get an anemia. It kind of works like that. So that's kind of what it's discussing here. Decreased globin production leads to imbalanced globin chain synthesis, defective hemoglobin production, and it can even be damaging to the red cells. So the way that thalassemias work, there's going to be two major types. There's alpha thalassemia and beta. They are named according to what chain is basically missing or absent, is not being produced enough of. So thalassemias tend to occur in the same kind of heritage as what we just discussed with sickle cell. Mediterranean through Middle East, India, Southeast Asia, south through Africa. So that same area of the world that we discussed with sickle cell and with malaria. So that's kind of, it says it seems to follow where malaria is prevalent. So just the thought there. There's two theories of why thalassemia, again, has kind of that resistance similarity, like we said with sickle cell anemia. One, it doesn't allow the parasite to really grow and thrive in the red cell, and two, it's increased phagocytosis. So my thought is it's just evolutionary. If you have these sickle cell anemia and thalassemia that are in the same area as malaria, it's going to be through time that you can revolve to have a resistance. So, all right, evolve, not revolve. Ugh. Anyway, so moving on. All right, so overall, the alpha genes are going to be on chromosome 16, and the beta genes that make the beta chains are on chromosome 11. So thalassemia will come from a decreased production of one of those, either the alpha or the beta. and then that will create an imbalance of the globin chains. Thalassemias are microcytic hypochromic anemias. So they are in the microhypo category. Most of the hemoglobinopathies from the last PowerPoint were normocytic normochromic. Like I had said, sickle cell anemia is definitely normocytic normochromic. The only one out of the last PowerPoint that would have been microcytic hypochromic would be hemoglobin E. Hemoglobin E is microhypo. The rest in the last PowerPoint were normocytic normochromic. So thalassemia is definitely microhypo. All right, so I'm actually going to skip this slide. I keep meaning to delete it, and I keep forgetting, obviously. You can read through it. It's good information, but we're going to basically get to the point of it here in a second. All right, so I want to reorganize how I kind of have this laid out. I'm going to get to this information. Don't worry. I know I'm skipping it for now, but we will definitely get to it, so don't worry. All right, let's start with the beta thalassemias. Again, named for that it's affecting the beta chain. So there's three categories of beta thalassemia. We have beta thal minor, beta thal intermediate, and then on the next slide it's going to be beta thalassemia major. So beta thalassemia minor is more of a heterozygous form of the disease, so it's a milder hemolytic process. And the hemoglobin is just slightly anemic, 10 to 13. There are some target cells and some elliptocytes. You're going to see, again, target cells throughout on these. Beta thalassemia intermedia is just kind of the next step down, a little bit more significant, in about a hemoglobin of 7, and then you have your big bad homozygous form of the disease called beta thalassemia major. Very severe anemia. It's often transfusion dependent. Their hemoglobin is sitting around 3 to 4, so they need lots and lots and lots of transfusions throughout their life to try to maintain even a closer semblance of a normal hemoglobin. So you will see hepatosplenomegaly here, jaundice, marked bone changes. So it's not just the globin chains. It does affect more than just that. You know, when they're making these globin chains in the bone marrow, there is impact to their bones. It will cause bone changes. There's impact to their spleen as these, red cells filter through and things like that. So there are more levels of involvement. Lots of poikilocytes, so definitely those target cells, teardrops, elliptocytes, red cell fragments, nuclear red cells, high amount of retics because it is a hemolytic process. These cells are very defective. When you can't make your beta chains properly, it impacts the way you're making your cell. It impacts, how they perform, if they're hemolyzed, things like that. So lots of stuff going on, but that's beta thalassemia major. Now, obviously, before I get off this, when you think about what hemoglobin molecule had two beta chains, that would be hemoglobin A. If you can't make your beta chains, you're not really going to have much hemoglobin A. You're going to have more A2 and F because those didn't involve the beta chain. So you have to always keep in mind what hemoglobin molecules had what chains, which one would be affected. So that's probably the key. I mean, thalassemias can get kind of confusing to people. You need to remember what hemoglobins had what chains, then you will know what's affected. So if you know beta chains are out of play, and you get a test question, you can't have hemoglobin A if you don't have beta chains because it was made of 2 alpha, 2 beta. So, keep that in mind that you see a significant decrease in hemoglobin A or probably none at all, and then you just have the A2 and F left instead, and those would be more increased in number as a result because that's what's there. There is a slight kind of other form called hereditary persistence of fetal hemoglobin. The name says what it is. You have increased levels of hemoglobin F even into adult life. Remember, hemoglobin F is the big one at birth, and then as you get older, you lose it. You have less than 2% usually, but in this kind of condition, they maintain a higher number. It's asymptomatic. It doesn't really cause them a huge problem. Just a quick note there. There's nothing else really I'm going to worry about on this one. All right, now let's go over to the other category, which are the alpha thalassemias. Now, there's going to be 4 sets of alpha thalassemias. And so first, before I get into it, think of it this way. When we talked about your hemoglobins and what chains they're made up of, every single one of your hemoglobins had 2 alpha chains in it, right? Hemoglobin A was 2 alpha, 2 beta. Hemoglobin A2 was 2 alpha, 2 delta. Hemoglobin F was 2 alpha, 2 gamma. So if you lose enough of the alpha production, it's going to really affect you. So we'll get into that last one, which is the most significant here in a second. So the first state is a silent carrier. You're only missing one out of the 4 globin genes at this point. So it's a carrier. There's no abnormalities hematology-wise. Person probably doesn't even know it. So they're only missing 1 globin gene. They still have 3 functional globin genes to make alpha, so they're still making alpha. Alpha thalassemia trait is now missing 2 out of the alpha genes. So they only have 2 left. So it's starting to be a little bit more of a mild anemia, microhypo. Hemoglobin H is the 3rd step. They call it hemoglobin H. You are now missing 3 out of the 4 alpha genes. So you only have 1 left to make chains. So what hemoglobin H is basically, it's unpaired beta chains that kind of formed the 4 together. They put 4 beta chains together. Since they didn't really have any alpha chains to put with it, they put 4 beta chains together, and that's what made hemoglobin H. So in this hemoglobin H disease, because they're only got 1 gene making alpha chains, they have hardly any alpha chains out there, so they're putting together the beta chains instead, and that's what hemoglobin H is. So you will see a lot of hemoglobin H in this, you know, if you were to do the electrophoresis, you'd see a lot of hemoglobin H to kind of hallmark it as this one. So it's still a hemolytic process, so you'll still get the increased reticulocytes and target cells and so on. The other thing to note here is it talks about hemoglobin H can precipitate and form Heinz-like bodies as well. And then finally, the big severe one is hydrops fatalis. There are no alpha chains being made. All 4 are gone for genes. As a result, you are not going to make any hemoglobins. You can't make A, A2, or F. It's like it says here, it's incompatible with life. So the baby won't even be born. It will be a stillborn, miscarried, whatever you want to say. It will survive maybe until the 3rd trimester, because remember, back to that hemoglobin chart, there were some other hemoglobins like Gower and Portland involved that didn't need hemoglobin A, or not hemoglobin, that didn't need the alpha chains. But the minute it's supposed to switch to making hemoglobin F, it can't. There's no alpha chains. And then that's when the baby will die. So it won't even be born. And it talks about here the predominant hemoglobin is hemoglobin BART. So what hemoglobin BART is, I'm going to go back to one of those slides I skipped. It's this one right here. Hemoglobin BART is 4 gamma chains put together. Because there was no alpha chains to pair with, the 4 gamma chains stuck to each other. So you do need to know that's what hemoglobin BART is 4 gamma chains. With that being said, if you go down to one of the last bullets, it says hemoglobin BART has high affinity for oxygen and will not deliver it to the tissues. So if the baby, and now we go back to where I left off, if you only have hemoglobin BART in that 3rd trimester because you couldn't make F or anything else, and that hemoglobin BART will not deliver oxygen, that's what results in the baby dying. There's no oxygen to any of the tissues. So that's what hydrops fatalis is, that 4th category of alpha thalassemia. All right, so those are the 2 big categories. Now, there are other thalassemias. There's combinations with different hemoglobins, like it shows here. You can have any one of those other hemoglobins combined up. I'm not going to test you on all this. This is where it gets really tricky and where it can become overwhelming. So it's just a note that, yes, you can have a combo of kind of anything. Overall, again, it's microcytic hypochromic, so you'll definitely see that, low MCV, low MCHC, increased RDW representing the anisocytosis. You get decreased hemoglobin hematocrit because it is anemia. In the bone marrow, you would see lots of cells trying to get made, but they're just kind of defective. They don't work well, so it just depends. Again, electrophoresis is a major tool on how we diagnose these. So we want to see what types of hemoglobin do they have, how much, you know, to know what's happening with that person. The Menser index is just a way to help separate a thalassemia from iron deficiency. Because iron deficiency is also microcytic hypochromic, has target cells, has elliptocytes, sometimes they can look similar if it's not the big, bad, major thalassemias. If they're more the minor thalassemias, they can look similar. So they can do this Menser index to kind of separate between them. It's not something I'm going to test you on. It might be on a study guide, but I'm not going to put it on a test. So if you don't want to remember it for a test, don't worry about it. I'm not that concerned on this part. All right, so that is it. I know these are not that exciting of chapters, I think they're confusing. They get to a deep, deep level of understanding, genetics and things like that. But, just do what you can with the major diseases in each chapter and the characteristics of each one. You have to get to know those hemoglobin molecules and what chains make each one up. That will really help you. So just do what you can. Use your study guide to help you. Definitely do your lab review questions. That also is helpful too. So if you guys have any questions, as always, please feel free to reach out. Otherwise, have a fabulous week. Thank you.